Hi and welcome back, guys. Grade 11 Life Sciences. Today we're looking at respiration. Let's see what we need to focus on. First of all, in terms of respiration, we look at the definition. The first and foremost thing is we need to ask ourselves, what is this word cellular, or these words cellular respiration, and what do they mean? Next up, we look at the process itself, glycolysis, Krebs cycle, and oxidative phosphorylation in aerobic respiration. So you'll notice we're talking about something there, aerobic respiration. We'll just hold that thought. Then we look at the definition of anaerobic respiration. That's why I asked you to hold that thought. You're talking about aerobic and anaerobic. So already in your mind you must know there are two different ideas here in terms of respiration, aerobic respiration and anaerobic. We need to find out what's the difference between these two. Further, we look at alcoholic fermentation and lactic acid fermentation. We'll explain those things later, so it's no sense going into details now. The role of anaerobic respiration in the industry. How does industry use this process of anaerobic respiration? Lastly, we want to compare aerobic respiration and anaerobic respiration. And because you have also studied photosynthesis before this, we would also want to compare the whole process of respiration with that of photosynthesis in between. Remember, some things you do not have to have a table in your textbook or in your study guide to say this is the difference between this process and that process. You've learned one process, you learn another process, you must be able to see the differences. Look at the differences in terms of where does it take place, what is needed for that process, how that process unfolds, and also what are the products of that particular process. So if you keep that in mind when you're looking at processes, any process, you ask yourself those questions, and based on those questions, you make your table of differences. It's as easy as that, guys. You all must be knowing Popeye the Sailor. We're talking about respiration, the process. The first question I ask is, why do organisms, yes, that includes you and me, and the ant crawling on your bathroom floor, and the bird singing outside, and believe it or not, all the plants in your garden, each one of every living organism has to respire. The question I'm asking is why? Why do they have to respire? And the clue is given in the diagram. You know, Popeye, I hope you know that this is spinach, very healthy for iron, and that is why they portray Popeye always taking the spinach every time he needs to get hold of Brutus and give it to him, give him a good beating or whatever, and he'll use the spinach. Why? Because spinach is associated with iron, and iron will help with hemoglobin, formation of hemoglobin. Hemoglobin is important to transport oxygen. So can you see, even in the cartoon, life science is there. So again, and you must have picked up the answer already, all living things need energy. Every living creature needs energy, whether it's a plant or an animal, whether it's a small plant or a small animal, or a large animal or a large plant. It doesn't make a difference. They all need energy. And the energy comes in a form of food, and this is equal to chemical energy. This food that we're talking about is in a big form. But when a cell needs to use food, it needs the food in the form of ATP. And when we discussed photosynthesis prior to this, we spoke about how ATP is formed. But we will look at it again. And here's the statement. Cells recycle ATP. What the hell does that mean? Let's look at this diagram. Here's ADP. ADP would be meaning A, P, and another one P. ADP. Adenosine diphosphate. Adenosine diphosphate. Why? Because there are two phosphates there. Now, this one can be combined with another P to form ATP. At the same time, this last P there, is associated with the energy. And when this P is given off there, the energy is released for cellular work, and the P from there is lost. It will form ADP. ADP plus P to give you ATP. Energy from catabolism. If energy comes in somewhere, ADP plus P will give you ATP, and so we can make that arrow there. ADP plus P gives you ATP. ATP, if the P goes away, becomes ADP. This is how ATP is recycled. Where does this process of respiration take place? Obviously, you know this by now, the mitochondria. Now we need to go into the mitochondria. Your grade 10 work coming to the fore again. If you look at the mitochondria, you'll find that it has an outer membrane, there, an outer membrane, and it has an inner membrane, and this inner membrane is folded. There you see the folds. That's the inner membrane. This area between the outer membrane and the inner, the outer membrane there and the inner membrane there, there's an area there, is called the matrix. Or they call it sometimes the ground substance. Question I have for you, and again guys, whenever you learn a structure, just now we spoke about when you're learning about processes, now a skill on when you're learning a structure. You need to know the diagram of that structure. You must be able to label the diagram, and when you label that diagram, you must be able to then also talk about how the parts that are labeled are suited for the function. So the question then goes, explain how this organelle is structurally suited for respiration. Now, let's first analyze the whole thing. Explain is a verb. And when they use the word explain, they mean you must go into detail. You must have some explanation into it. For lack of a better word. You need some, or let me use a better word, you need some elaboration in this thing. So how is this organelle structurally suited for this? For this, we must look at the structure. We must consider in our mind the function, and we must be able to marry these two things, the structure to the function. And let's look at that. First of all, it has a double membrane, which is permeable to gases and water. So it has a property of permeability. And what does this do? It allows gases and water to pass into or out of it, which is important because it's involved in cellular respiration where gases are involved, where water vapor is involved. So you must be able to enter or leave, whichever way they're going. Also, the inner membrane is folded. And anything in life science, anything in the body, any structure in life science or biology that is folded increases the surface area. For what? For attachment of respiratory enzymes. So what does this actually mean to you and me, the ordinary guys? It simply means if this membrane is folded, it is larger than what it looks like. And if it is larger than that, what does that mean? It means it can attach more respiratory enzymes. And if it can attach more respiratory enzymes, that means more respiration can take place. So it is geared, its main function, remember mitochondria in grade 10, you said it's the powerhouse of the cell. Why? Because it is involved in cellular respiration, which releases energy that is necessary for work. So how does that happen? This whole story is taking place, and with respiratory enzymes taking their place in this area, more space, more respiration. Good. So now again, we're going back to what is respiration. We asked earlier, why does respiration take place? We then asked the question, where does respiration take place? Now we're asking, what is this respiration that we are talking about? Remember, in our first opening statement, we said we need to know the definition of respiration. And there we are now there. First of all, cellular respiration is the process, we know that already, that releases energy. How? By breaking down food molecules in the presence of oxygen. These food molecules that we are talking about are organic molecules. You learned this in grade 10 as well. Organic molecules. Carbohydrates, fats, proteins, etc. Organic compounds. Mainly in respiration we would talk about glucose, but remember, lipids have the potential of becoming carbohydrates. Proteins have the potential of becoming carbohydrates when they are converted. So all of them, all organic molecules can be used for cellular respiration. Now that is the word explanation, is a definition for cellular respiration. The next thing we need to ask ourselves, so how do we put this as an equation? You know, we're doing science, life sciences. We're not just doing anything, we're doing life sciences. So there's a science involved in what we're talking about. So we talk about how can we put this, we're talking of a process, a chemical process. This chemical process has gone from X to Y to Z or whatever it's doing. There must be a formula for that. And hence, we then put the formula down for you. Ignore the numbers first. O2 plus C6H12O6, that is glucose, will give us carbon dioxide, water, and energy. That's what we're getting there. Now, why do we put these numbers here? Yes, I can see all the science students, the physical science students, all putting your hands up. Why? Because you know that if we just left it without the numbers, the equation would not be balanced. So to balance the equation, we had to add in the numbers to get there. And this is not the forum for going too deep into that right now, because others will get confused. So if you know that, good. But remember, it's a balanced equation, that's why it's there. Now, that's a formula, a chemical formula. Now we want the same equation in words. In words. And how do we do that? We simply say oxygen. Notice now we don't have to balance because we're just talking in general terms. We're not saying how many and what have you. Oxygen plus glucose, that's on the one side, gives us carbon dioxide, water, and energy. So if I asked you, what are the requirements for respiration? From this definition and from these equations, what would you be able to say? What are the requirements? The requirements would generally be on the left-hand side of this equation, yeah? Definitely. So the requirements are oxygen and glucose, or oxygen and organic compounds. What are the products of cellular respiration? Carbon dioxide, water, and energy. The carbon dioxide, the water, are actually byproducts. They by the way, and they have to be released from the body somehow. We'll talk about that a little later. The main purpose of respiration is to release the energy. This energy, by the way, is released in a form of ATP. And that is how cells get spending in terms of energy. ATP is a unit of energy for a cell. Now let's go a little deeper. Before we go there, let's have a light-hearted moment. First of all, let's get a definition again. Cellular respiration, process in which sugars, glucose, are converted into usable energy, ATP, in the presence of oxygen. Later, you're going to tell me, yeah, but you told us in the presence of oxygen. In the presence of oxygen, every time you use this. Yes, I'm using that because that's the general way. But later, we're going to learn something, and I'll remind you about what I'm saying now. Now here we're talking about, look at Mr. Simpson there. You know Mr. Simpson? Yeah. He's having the sugar in the form of a donut now. Remember, he's eating that, and when he eats this donut, the donut represents mainly carbohydrates. And when he digests that, it's going to eventually get digested to glucose. So, there's the sugar there, the glucose, plus the oxygen. It's going to come from, where the oxygen come from? Did he eat the oxygen? No, 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 no. Came from gaseous exchange, from breathing it in from the air. Oxygen is around us. So we're breathing in this oxygen, and when it gets to the lungs, gaseous exchange takes place. The oxygen gets into the blood, eventually gets distributed to all parts of the body. So that's where the oxygen comes from. So the sugar plus the oxygen gives us carbon dioxide plus water plus energy. Notice that we have a cartoon here, but our word formula hasn't really changed. We may have said sugar instead of glucose. That's another term that is used. But the general trend in what's happening here has not changed. Now the next question is carrying on with what is respiration. Now we have to take you a little bit back before we move forward here. And remember, we started when we spoke about photosynthesis that the radiant energy comes from the sun, and these guys in the green plants called chloroplasts, they trap this radiant energy in photosynthesis and they make organic molecules. So they are manufacturing organic compounds, and therefore that's an anabolic process because it is making food. Whereas, look at what happens during respiration now. Respiration, these organic molecules are used in the mitochondria to release energy, to take the energy that is trapped in this organic molecule and release it in a form of ATP, and by the way, release carbon dioxide and water. By the way, if you remember your photosynthesis, you'll know that the carbon dioxide and water are both necessary for photosynthesis. So this diagram is showing you how in nature the gases are recycled. One is the plant is using the carbon dioxide and water during photosynthesis. Remember, the plant also respires. Don't forget that. The plant also respires, and for respiration it still needs oxygen. I'm going to repeat what I said on photosynthesis, that get out of the myth that plants take in carbon dioxide and animals take in oxygen. It's got nothing to do with plants and animals. It's got to do with processes. During respiration, oxygen is used, carbon dioxide is released, and water vapor. During photosynthesis, carbon dioxide and water are used, and oxygen is released. What wonder creation we have. That all these things were put in there for us. Each thing has its function. And as things are being used and released, others are used from other organisms. And so the cycle continues. Each thing in its place. Imagine the small little thing called a chloroplast, which you and I can't see with our naked eyes. The small thing called mitochondria that you and I cannot see with the naked eyes. But massively complicated processes take place in these structures to give us the necessary energy and sustenance that we need. All was put in there before the computer was invented. Okay, good. Now we're going to the how. The next series of slides will talk about how does this respiration take place. We've spoken about what is respiration, where does it take place, why does it take place. Now we're talking about how it takes place. Basically, there are 3 stages in cellular respiration, and that is, we talk about aerobic. That means if the complete respiration takes place, there are 3 stages. The first stage is called glycolysis, then follows Krebs cycle. Some textbooks will call it citric acid cycle. And then follows the electron transport chain, or what we named earlier as oxidative phosphorylation. Others may even call it the hydrogen transfer system. So those are the three areas: glycolysis, Krebs cycle, and the oxidative phosphorylation or electron transport or hydrogen transfer system. Let's take each of these and explain them now. Let's start with glycolysis, the first phase. Where does this glycolysis take place? This glycolysis takes place in the cytoplasm of the cell. You must be able to say where it takes place. What happens during glycolysis? The glucose is broken down into a compound called pyruvic acid. And while the glucose is broken down, energy-rich hydrogens are given off in that time. The hydrogen move into the mitochondria to be used in oxidative phosphorylation. We'll talk about that later. It's not discussed here. We are talking about glycolysis. We're just telling you what will happen to the hydrogens later. During this phase, 2 ATP molecules are produced. So the end of glycolysis, if your respiration ends here, if it ends here, then you will only have produced from each glucose molecule that you consume or that you produce during digestion. You would only be able to get 2 ATP molecules. Now let me tell you something. 2 ATP molecules won't even get the ability for you to think to move this finger. Leave alone move the finger, but to think about moving the finger, these 2 ATP molecules are not enough. So for larger organisms like us, glycolysis on its own, not good enough. Let's go further. By the way, diagrammatically, this is what's happening. In the cytoplasm, this is all happening in the cytoplasm here. All this, cytoplasm, all of that in the cytoplasm. What has happened? The glucose is broken down to pyruvic acid. There's the glucose. It is broken to pyruvic acid. While the glucose is converted to pyruvic, remember, it's not as straightforward like we're showing you here. There's much more detail in terms of what we're talking about. But for our study, for the curriculum purpose, this is what you need to know. If you want to read more, please do so. Okay? But for your exams, remember, this is what you need to know. So the glucose is converted to a series of reactions, obviously, into pyruvic acid or pyruvate. What has happened in that time? ADP is converted to ATP. So there's the ATP that we are talking about that has been produced. At the same time, NAD, a carrier, energy carrier, picks up the hydrogens that are released from the glucose and forms what we call NADH. So now, what are the products of glycolysis, if I have to ask you? What are the products of glycolysis? What do we mean, what are the products? What is this man, crazy man, white-bearded man, asking us what is the products? Is it a maths problem? No. Products in this context means what has been formed in this process. What's the end result? What have we got out of it? Yes, I see you smiling. That's the thing. You got ATP and you got hydrogens, which combined with NAD to form NADH. These are the products of the first phase. What you must know is what started? Glucose. What happened to glucose? Glucose was converted to pyruvic acid, and in this process, we released some hydrogen from the glucose and we released some phosphate from the glucose to make ATP from ADP. That means there was A, P, and another one P. And we added another P. We used some of the energy to use the P, to combine that to ADP to make ATP. Remember, when we move from ADP to ATP, we are making energy. We are creating the energy there that's called ATP. When we move from ATP to ADP, it means we are utilizing the energy. The phosphate is being used to release the energy that's contained in that phosphate. And that's why the ATP breaks up into ADP plus P. That's what you have to understand. Now, that being said, we're not going to look at the whole diagram there. We'll come back to that just now. The next phase is known as the Krebs cycle. What happens to Krebs cycle? What will be left with in glycolysis? Pyruvic acid, isn't it? The compound that was left was pyruvic acid. And the products that we had were ATP and NADH. Why NADH? Because hydrogen combined with the energy carrier NAD to form NADH. Now what happens? So the pyruvic acid is broken down into energy hydrogen and carbon dioxide. This pyruvic acid goes through this whole cycle in the Krebs cycle. Again, it's a complicated cyclic thing. There's a series of reactions that take place where the carbon and the hydrogens and the oxygens in the pyruvic acid are broken down, and the carbon dioxide is released, and the hydrogen again, they are going to go to, as we said in glycolysis, they're going to go to this thing that we call oxidative, not this thing, this process called oxidative phosphorylation. There we have it there. And the carbon dioxide will be released eventually from the lungs. Now we go to oxidative phosphorylation. Before we go there, let's go back to our diagram. We were there by pyruvic acid at the end of glycolysis. From there, we are looking on this side. Don't worry about the left-hand side of the diagram now. The pyruvic acid goes down, and in the cycle of reactions, ATP is produced again, and the hydrogen is released, and carbon dioxide is released. The carbon dioxide is released, it will go to your lungs, and from the lungs, it will be exhaled. This hydrogen is going to go to the next stage, still in the mitochondria, known as the oxidative phosphorylation. Okay? So we've looked at it in words, we've also looked at it in pictures. Oxidative phosphorylation. What happens here? The energy that was made from the energy-rich hydrogens is used to make up ATP. That ATP we saw in the last diagram, this ATP. The energy from the hydrogen is used in the hydrogen transfer system to make ATP from where? From ADP. Whenever they say that ATP is made, it means ATP was made from ADP plus P, gives you ATP. And during this process, by the way, during this process, 36 ATP molecules are made. So from the same glucose, one glucose, 2 from glycolysis, plus 36 from the end of Krebs cycle or oxidative phosphorylation, that gives you a total of 38. Now, where is 38 ATPs and where is 2? Can you see the difference if the second part of respiration does not take place? How little energy that you're going to get. And consider that when we're going to talk about something a little bit later. Now, listen to this part here. We said oxygen is necessary for this process. But watch, we've spoken about glycolysis. Did we mention oxygen? No, we didn't talk about oxygen. Oxygen was not used anywhere there. We spoke about the Krebs cycle. Did we speak about oxygen? No, nothing happened there. Now we're talking about oxidative phosphorylation. Let's see what happens. The energy depleted hydrogens. Remember, the hydrogens started in glycolysis. They were released in glycolysis. Some more hydrogens were released in Krebs cycle. They were picked up by NADH, and from there they went to this oxidative phosphorylation. And what has happened? They combine after they get tired. In inverted commas, tired. You can't write in exams that the hydrogens get tired. Okay? The energy, when I say tired, I'm trying to make you understand that they are depleted of energy. They are energyless now. All the energy that was there, the process has used up. to make ATP. So this energy depleted hydrogen combines with oxygen. Here comes the oxygen now only. Oxygen is only here to make water. And remember, we said earlier that in our formula, water is part of the reaction at the end, or the equation. This water is then gone back into the system and can be either given off as water vapor during exhalation, or it could be filtered through the kidneys and be excreted in the urine as water, excess water as well. Okay? Water vapor from the lungs when we breathe out, or it could be excreted with the urine. And also some of the water we can lose in the sweating as well, remember that. Now guys, what happens if this oxygen is not there? Then all of this is wasted. It doesn't take place if that oxygen is not there. So the whole Krebs cycle and oxidative phosphorylation will not take place. Something will happen to the pyruvic acid that will not follow this pathway. So can you see, the oxygen only comes right at the end, but if it's not there, none of these things can happen, and you'll end with the 2 ATPs there. Now a summary. A simple summary of what we've been talking about all the time. Guys, and the idea of putting these slides here is to form a mind map in your mind. We've done all this work here. Make some sense of the work. Now, don't memorize everything. Just make sense. Okay, we've done this. What has happened there? Follow it through in a systemic, logical sequence. And you'll see how much more you're going to remember your work, because you're learning to understand. You're not learning to forget. Most of us learn to forget. We learn for the test. After the test, we've forgotten what we've learned for the test in the first place. Learn to remember. But first, if you want to remember, you need to understand. Because once you understand, you will make meaning to it. If somebody tells you, talks to you in another language, and if you don't understand that language, what sense, if you give you whole instructions of where to find gold, but he's talking to you in a language that's foreign to you, how are you going to find the gold? No chance. But if you understood what he told you, you could maybe try to at least get the clues and so on. Huh? All right, so let's see what we're talking about here. First and foremost, glycolysis takes place in the cytoplasm. What has happened in glycolysis? Glucose is broken down to pyruvic acid, and ATP molecules are formed, and some hydrogen is released. This hydrogen combines with NAD to form NADH. The pyruvic acid is involved then in a series of reactions in the Krebs cycle, where carbon dioxide is released and hydrogens are released. This hydrogen is going to do the same as the other hydrogen to form NAD. The hydrogen, the energy from the hydrogen is used in the next phase called electron or hydrogen transfer system, or what we are calling it, oxidative phosphorylation. Phosphorylation. That should be a Y. Okay? Notice there 36 ATP molecules. 2 from there plus that will give you 38. Also notice that water is given off there. And this is where the oxygen comes in to this phase here. By the way, this word oxidative phosphorylation, a big intimidating word. Oxidative, by burning something, oxidation is taking place, using oxygen obviously. Phosphorylation, adding phosphate. We had ADP plus P, phosphorylation, to get ATP. So understand the word. Every day you're going to say oxidative phosphorylation, oxidative phosphorylation, oxidative phosphorylation, and you don't know what it means. Meaningless, senseless, no significance. You're not going to remember it. But if you understand what it means, and how do you do this, guys? Even though somebody may not explain it to you, how do you get past this? You have a textbook, right? I'm hoping you discovered by now, at the end of your textbook, there's a thing called glossary. So anything that's new to you, the minute you hear a new word, turn to the glossary and make meaning of that word. Write it down if you have to. Don't wait for the end of the year to learn 100 terms, when you can learn 10 terms per topic. 10 terms per topic. In that way, you learn all your terminology. Your section A becomes easy for you at the end of the year as well, and your definitions become easy for you. So this is where we are in terms of the process. This is how this process has taken place. I'm hoping that we have taken in as much as we could while the explanations were taking place, and I'm sure by now your mouths are watering. You may be jumping on the lounge suite there, needing to go to the toilet. So let's take a quick break. Hi guys, and welcome back. I'm hoping that you've enjoyed your break. You've used it wisely, had enough liquid to keep you sustained for the next half of this segment today. Now, if you remember, as we were talking, we were mentioning this word aerobic, aerobic respiration all the time. And at the beginning, I told you to hold on to that thought. And you notice I said to you just now that if oxygen was not present, then Krebs cycle and oxidative phosphorylation could not continue. It would stop by glycolysis and take another pathway from there. Now, before we actually talk about the next idea, let's talk about these two things quickly. Aerobic and aerobic. These are the two words that we have. Aerobic, oxygen is necessary. And anaerobic, oxygen is not necessary. In fact, anaerobic is without oxygen. And by now, if you're a life science student in grade 11, you should have picked this up, that in biology, anything related to biology, and we calling it life science, anything in this subject where the words or the prefix an or a are added to a word, it generally means without or not. For example, or none even. For example, in grade 10 you learnt about the components of an ecosystem, and you classified them as biotic, and biotic were all the living organisms, and abiotic. So you put the A in front of biotic to say non-living. Cellular, you learnt earlier in the year, cellular and acellular. Viruses, acellular. What did that mean? It's not a true cell. Okay? So remember that as well. So anaerobic is without oxygen. So keep that in mind as we go along. Now let's look at what we're actually talking about there. Anaerobic respiration. So the question is, what happens if oxygen does not reach the mitochondria? Remember we said, let's go back. We always go back to our slides. There's the oxygen there. Came into the mitochondria for oxidative phosphorylation to take place. Now what happens if this oxygen did not reach there? Anaerobic respiration occurs. So glucose will still be broken down, but if there's no oxygen, then anaerobic respiration will take place. So what are we saying? Let's look at a diagram. Here's the glucose. We're starting there. So notice the label there, glycolysis. Glycolysis. During glycolysis, nothing changes. Glucose is converted to pyruvic acid. So glycolysis will take place. Remember, I stressed that point that at the end of glycolysis, only 2 ATP molecules. So that's what we're bringing back in now. So if there's no available oxygen, and if it's a bacteria and some mammals and animals, then the pyruvic acid doesn't go through Krebs cycle. It doesn't go through oxidative phosphorylation. It is used in reactions to produce what we call lactic acid. And this process is called lactic acid fermentation. So it's in animals, when respiration takes place and there's a lack of oxygen or there's no oxygen, then the pyruvic acid is converted to lactic acid. This is known as lactic acid fermentation. Simple. Don't go into too much detail. That's enough you need to know. We're going to talk about interesting stuff just now about that. But this is what you need to know for curriculum. In a plant, if, like for example, yeast, if there's no oxygen, the pyruvic acid is converted to ethanol and carbon dioxide. This carbon dioxide, by the way, is used in the baking industry. And ethanol, you can hear the word ethanol, alcohol, and this is known as alcoholic fermentation. Alcoholic or alcohol, rather let's use the word alcohol, fermentation. Sometimes when we use this word alcoholic, then the first thing that goes in a child's mind is drunk. Alcoholic, drunk. And 9 out of 10, when we ask these questions in the exams, and we talk about yeast and we talk about alcohol, lots of children give the answer that the yeast is drunk. Now, how ridiculous is that? The yeast is drunk. Think about it. Now, I said we're going to talk a little bit of interesting stuff. Yeah, look at this guy. He is fatigued. He is fatigued. What is that actually? Fatigue, not fatigue. It's fatigue. In the Indian dialect, Indian language, fatigue means to be torn. And that's how he looks. He looks like he's torn. Fatigue. In this case, what has happened? He's been running or whatever he's doing, but he's very tired. Why? Because the oxygen is not reaching the cells where it's needed, the muscles and the cells where it is necessary. So therefore, what has happened in his body? The pyruvic acid is converted to lactic acid. This lactic acid fermentation that takes place in muscles mainly causes cramps. And you know that you have a burning sensation when you have cramps. Now, how would we put this in a formula or an equation? Equation, better word, C6H12O6, that is glucose, is broken down without oxygen. Yeah, there's no oxygen. We're not saying oxygen plus. It's broken down into alcohol and carbon dioxide and the ATP that came from glycolysis. 2 only. Little bit of energy, alcohol and carbon dioxide. That's mainly in plants. Alcoholic fermentation. Okay. In words, glucose is broken down into alcohol plus carbon dioxide plus energy. You need to know both the numerical representation or the scientific equation and the word equation. And in animals, like humans, C6H12O6, the starting point is the same, doesn't change. In all, even in aerobic respiration, the starting point is still glucose. What happens to this glucose? It is converted into 2C3H6O3, which is lactic acid, and 2-ATP, which is the energy, represents the energy. So this is how you would represent this particular concept. So what have we done so far? We've looked at aerobic respiration, which is made up of glycolysis, Krebs cycle, and oxidative phosphorylation. And now we're looking about if oxygen is not there, what happens? We looked at two things. One is alcoholic fermentation. That means where pyruvic acid is converted to ethanol plus carbon dioxide plus ATP, or lactic acid fermentation, which is pyruvic acid converted to lactic acid plus ATP. Notice there's no carbon dioxide in this process that we are talking about here. Good. Now comes my favorite part, and that is we've done so much of work and you've done work prior to this. how do you actually get there? This what we do is so much more important, guys. And this you must get into the practice of doing at school. You're going to do a chapter at school, go home, look for questions in your textbook, or find, the internet has plenty questions now. Get a past paper and go over that section. That is going to tell you what you have learned to understand and to remember. That which you don't know, you're going to go back the next day, tell the teacher, hello, sorry, this part is a bit grey for me, please explain. And that's how you give feedback to us when we are teaching you. So first question says, why is respiration important to organisms? And if you remember, that was our first question when we started the show. First of all, we spoke about, obviously it gives, or we'll come to that now, it maintains the oxygen balance in the atmosphere. Because if the oxygen was just left in the atmosphere and organisms did not use the oxygen for respiration, there would be too much oxygen in the air. So in this way, the organisms that use oxygen for respiration are maintaining the balance. At the same time, it also maintains the carbon dioxide balance. If plants used up all the carbon dioxide for photosynthesis and there was nothing releasing the carbon dioxide back into the environment, there would be no carbon dioxide for the plant. So, during respiration, what do? We exhale carbon dioxide. We produce and exhale carbon dioxide. So, we release carbon dioxide to the atmosphere. The plants can then use that carbon dioxide for their processes. More importantly, and this was our first answer, why do we respire? Remember, this question goes a little further. That question said, why do we respire? This one saying, why is it important to organisms? So you need to go further. Okay? And the last one is what we spoke. It must provide energy to heterotrophs. Again, now, if you're sitting there scratching your head and asking yourself, what in heaven's name is a heterotroph? You are in serious problem. Because you should know this word. But even if you don't, you're not going to die because you don't know this word today. Take your textbook out, look for the word heterotroph, and see what the meaning is. But I'm going to explain it anyway. But what I'm saying is when you normally have the situation, that's what you should do. Heterotroph. Troph means feeding level. Always nice to break the word down. Feeding level. Hetero is opposite, not opposite, is different from auto. Auto would mean self, and we put them together, self-feeding, meaning they manufacture their own food. Hetero cannot manufacture their own food. That means all the organisms in an ecosystem that are dependent directly or indirectly on plants are known as heterotrophs. So this is why we need to have respiration. Next question. Draw a label diagram to illustrate the general structure of mitochondria. And remember in life science, guys, they're not going to tell you draw a diagram without labels. So even if the diagram, if the question sometimes maybe they made a mistake not putting labels, you must label diagrams. If you do not label, there's no marks. So there's no such thing as drawing a diagram without labels in life science. So whenever you have to draw a diagram, remember you must have a caption, a heading, and you must have labels. And some general rules about that as well. And we're just putting a picture there, but I'm going to show you that it's very easy to draw this as well. That's what a mitochondria should look like. And there's the heading, and there's the diagram, and so on. But now what if you had to draw that yourself? And you're saying, yeah, this guy thinks he's smart, but he got a big diagram from the computer, and he can't draw to save his life. Okay? So let's see if we can beat that test. Mitochondria, look at this, how easy it is. Notice that when I put the pen down, I went right round. And this is on the screen. On your book, it's easier. I didn't lift the pen and start shading. So that's one golden rule. So we drew one membrane. That's the outer membrane. Now we have to worry about the inner membrane. And what do we know? The inner membrane is folded. There we're making all the folds. And there you have the mitochondria. So your heading, mitochondria, your caption, underline it, and then you start labeling. The thing about labeling, You can label anywhere in a diagram. You don't have to label in a straight line on the left or in a straight line on the right. There's no such a rule. The rule is your label lines must not cross. Okay? So if you're labeling this as the outer membrane, you can't put another label line going across there. Okay? It mustn't cross. It must go elsewhere. So you can go there. This is the inner membrane, or called crista. More than one cristae. And this is the matrix. Or some textbooks prefer to call it the ground substance. And that's all there is to drawing a mitochondria and labeling the mitochondria. There's nothing else. If you want to be smart, you can put in a little thingy there, but you can't see that generally, so I wouldn't even show you how to do it. Leave that out. Leave that out. Just these are the labels that you need to know. I'm hoping that helps you. Good. Now we're going to the heavier questions. This one was taken from a 2010 standard grade paper from NATED. Remember grade 11s? If you want to look for resources, the NATED papers, paper 1 and 2, are relevant to your syllabus. So you can look at very old papers and you can use that. So study the diagram below and answer the questions that follow. Simple instruction. Here's a diagram. They're telling you here there's two test tubes. Yeah, there's two test tubes here. In the one test tube, we have a rubber stopper there. Inside we have a solution with yeast and glucose. Yeast and glucose, important to note that. Going out of this tube, or rather of this test tube, is a tube, glass tube there, going out there, going across, and into another solution in the next test tube next to it. This one also has a rubber stopper, and it also has another tube going out that way. Here we're saying there's a liquid X, some liquid is in this container here, and there are some gas bubbles there. Some gas bubbles that we are finding in this container. And they're saying apparatus to investigate a biochemical process. Now obviously we're dealing with respiration, so it becomes easier. But in an exam, it's a whole paper on the whole syllabus, so you need to know which biochemical process. So it says state one function of the rubber stopper in tube A. There's a rubber stopper and we want to know what the function is. And the function is to make it airtight. To make it airtight. And what does that mean? It prevents atmospheric air from entering or escaping from this solution. The only way that whatever comes out from here, any gas that comes out from here, it must only be able to go into this tube, nowhere else. We mustn't lose the gases. So airtight. Airtight meaning that you are preventing air from coming in or going out. Airtight. All right? Next question says, name the biochemical process that is taking place in tube A. And we spoke about that already, but you must be careful because yeast is mentioned. You should know that yeast is undergoing anaerobic respiration. If you gave the answer cellular respiration, it was marked wrong. Because yeast is not just cellular respiration, it's a particular type, and it's anaerobic. Or we can talk about fermentation. And if you want to be more fancy, you can say alcoholic or alcohol or alcoholic fermentation. Then they want to know the gas bubbles in test tube B would be carbon dioxide, because during anaerobic respiration in yeast, carbon dioxide is released. The liquid in tube B can either be, and thus you had to know your experiment, you needed to talk about this experiment or have been demonstrated to or have done the experiment to understand it. And the liquid is lime water, or sometimes people may use bromoethyl blue. Okay, just by the way, these are indicators and they indicate the presence of carbon dioxide. Okay, name 3 products formed in the biochemical process that is taking place in tube A. Remember, this was anaerobic respiration. And they want you to name 3 products. And I explained that earlier. Products are things that are produced or manufactured in this time. And they are, yep, you got it guessed right, alcohol, carbon dioxide, and ATP. You don't have to say how many ATP or whatever, but you got to say ATP there, because they didn't ask you exactly. Next question says, describe how you would set up a control for this investigation. Now, very important to understand the skill. The skill that is being tested here is that of setting up experiments. When you want to set up any experiment, be it an experiment in your own house, about your own, anything about yourself, about your eating habits, or about something that's happening, there's too many ants, what can you do about it, or whatever else you want to investigate. You cannot just investigate without having a control. A control is generally, you got to set up apparatus that is similar to what you've set up, but you leave out that which you're testing for. Everything else must be the same. Then it's a control. Okay? So how would you do it here? I've already given you the first part of the answer, and that is set up the apparatus as shown, but do not put yeast in test tube A. You see, the minute we take out yeast, because one can argue that carbon dioxide came from the glucose in the water. And we are saying that the carbon dioxide is coming from the anaerobic respiration. And where does the anaerobic respiration take place? In the water? The water respires? No. The sugar or the glucose respires? No. It's the yeast that's a living organism that respires anaerobically to produce carbon dioxide. So we take that guy out. We take the yeast out and we set up the same apparatus and we leave glucose and we leave water there. And we would have got similar results. Some may argue that if you do the same thing, you put yeast but you don't put glucose, you may get the same result, because without the food, the respiration cannot take place. Okay. Give one reason why the process illustrated in the diagram is economically important. What process was it? Anaerobic respiration. Why is it economically important? It helps in making alcohol, beer making, wine making, or produce alcohol, any one of that. And it helps in baking to form carbon dioxide. Remember, notice here, I like this instruction here. This is in the memo. This is what will come to the markers. Say mark the first one only. Why? Because the question said give one reason. If the question did not say give one reason, they said explain the economical importance. Even if you gave 10, they would have had to mark all 10. But if they say give one or give two, whether it's giving one or two, providing one or two, or saying tabulate the two differences, your first two are marked. After that, nothing is marked. So you must make sure that your first two, or your first one, or your first three, according to the question, is what you're talking about. State one reason why it would have been better to use a thermos flask instead of a test tube. A thermos flask is a flask that keeps the temperature in. It's a flask that you keep your tea or coffee, you want to keep it warm. That's a thermos flask. It keeps the heat in or heat out. Why would you do that? Because we want to keep the temperature constant. Remember that for respiration to take place, enzymes are important, and enzymes are sensitive to temperature. So if we fluctuate the temperature, then the enzymes will not produce, or rather they will not catalyze the reactions at the right rate. So that's why we need to keep that. Because remember, when we're testing, what are we testing for? Whether yeast gives off carbon dioxide, especially during cellular anaerobic respiration. And everything else that we're testing for must be kept constant. Good. So that was an investigative question. We move on to another investigative question. A lot of reading here. And now I want to show you the technique when you get such long questions like this. First of all, they're saying, and watch my pen and watch what I do with my pen. A group of learners investigated whether all organisms release carbon dioxide. So we already got the gist of something. We're going to talk about that just now. Now they go further to say, they set up the experiment as shown in the diagram below. All the test tubes were kept in the light, and they used an indicator to show the different concentrations of carbon dioxide. The following color changes of the indicator revealed the levels of carbon dioxide present. Normal carbon dioxide, indicator turns orange. Low amount of carbon dioxide, indicator turns purple. High amount, indicator turns yellow. What have I just done? While I was reading that, I have underlined the key words. So I start understanding where the experiment or where this passage is going. Now initially you will have to do it physically like I'm doing it here. But as you develop this technique, then you will make mental notes of the important words that you are looking for. Now they give you the diagram. There's the light. There's test tube A, test tube B, test tube C. Here they have fresh green leaves. Here they have a bag of worms. Here they have some air, and they have a carbon dioxide indicator. All of this, obviously, because they're looking the same, all these will be the carbon dioxide indicator. All of them got it. And obviously, all of that will be air. But you'll notice that there's nothing else in this particular, test tube. The table below shows the results. Now we're looking at the results. What are these results? In test tube A, initially orange, orange, orange. It was all orange. Test tube A, at the end, after a few hours, purple, yellow, that's yellow by the way, and orange. Obviously, what must we do? We have to go back because we can't remember what they said. They said if it's normal, then it's orange. Okay, we can go immediately here and start writing. Orange, N, normal. Low amount of carbon dioxide turns purple. Go back here. Purple, there we go, one purple there, low. And the other one is high, it's yellow. So already as you're going along, without answering a single question, you have made important progress in the question. Now we can start answering the questions. What is the purpose of test tube C in the investigation? We just dealt with a question similar. What have we done in C? We've put the same water and the indicator and air and everything else the same, but what have we done? We've put no organism in it. So what is that? We're removing that factor which we're testing for, so that would be, it acts as a control. It acts as a control, acts as a control. Why? To compare the results. Again, now a famous question they like to ask, what is the purpose of a control? A control is there so you can compare. If you don't have something to compare, how are you going to know what you're doing is what's causing the difference? You get up in the morning, you have some eggs, you have some polony, and you have guava juice. Immediately after that, you start throwing up. Now, does it mean tomorrow you mustn't eat guava juice, you mustn't drink guava juice, you mustn't have eggs, and you mustn't have polony? How are you going to know which of those three things caused your problem? Or if indeed, if any one of them caused your problem, maybe something else that caused the nausea. So what do? You start leaving out one at a time. You leave out the guava juice, you have the other two. If you're still nauseous after that, you know that it's not the guava juice. And so you carry on if you want to investigate that. So this is what we're talking about, a control. Give one reason why it was necessary to seal each test tube with a rubber stopper. Again, guys, look at this. It's a different question altogether, but testing the same thing we tested just now. And because we've done it so close, it's easier now. So why? Obviously, to prevent movement of air between the test tubes and the atmosphere and vice versa. In other words, what's that big word we used just now? To make it airtight. And you must explain it because, or they say give one reason. They say reason, so you must give a reason. So airtight, why? Because of this. I would have given you the mark if you said airtight because that's what it means. What gas do you think would increase in concentration in test tube A? Explain your answer. You know, if I was you, if I was a pupil, no, let me not tell you that. That's not a good idea. What gas do you think would, just to tell you that this is not a nice question. Never ask the question, what do you think? You say, what gas would increase? Explain which gas would increase and so on. We prefer not to use this, what do you think? But anyway, that's a matter for another day. What gas do you think would increase in concentration in test tube A? Explain your answer. Let's go back to test tube A. Oops, it's on the next page. Test tube A, what have we got here? We got fresh green leaves and we got the solution. Fresh green leaves, sorry, and the solution. And what do we know about plants? That they're going to use CO2 for photosynthesis and they're going to use oxygen for cellular respiration, but when they photosynthesize, they're going to release oxygen. So keep that in mind. The green leaves undergo photosynthesis, they release oxygen. So our answer is oxygen, as I said. Why? Because they undergo photosynthesis to release the oxygen into test tube A. Explain the results of each test tube after a few hours of the investigation. Now you have to remember what they're talking about there. We're just going to go through the answers quickly. Firstly, in test tube A, photosynthesis took place and carbon dioxide is used up. So it has a low amount of carbon dioxide, and because of that, it caused the indicator to turn purple. So we know the result because the result was given to us, purple. So what have we done? We've worked backwards. Why did it become purple? Because there's a low amount of carbon dioxide. And the next question, why was there a low amount of carbon dioxide? Because photosynthesis uses up the carbon dioxide. You're working in a reverse. Now let's look at test tube B. What is the result? The result, it turns yellow. What does yellow tell us? That there's a high amount of carbon dioxide. Why is there a high amount of carbon dioxide? Because respiration is taking place more and carbon dioxide is given off during respiration, therefore it's high amount. Now the last test tube, C, the control that we spoke about. It remains orange. Why? Because it remained normal. Carbon dioxide is normal. And why? Because there was nothing happening in this test tube. There was no plant, there was no animal. So nothing changed, it remained the same, and therefore the color change given to you. Predict what color the indicator would be if the learner set up a fourth test tube with worms and leaves. Remember, what they got so far is leaves on their own in A, worms in B, and here they got nothing in C. So they're saying, let's say we had a D here, and in this D we put some leaves and a bag of worms. You put them both there and we have, so this, I'm drawing it just so that you visualize it, what's happening there. Okay, let's go to the question now. What's going to happen? First of all, there will be no color change. Generally, remember these are probable. We can't say for sure. But generally, should be no color change, or it remains the same, or it remains orange. Remember, no color change means remains orange. Because when we started with orange. And after a while. Now, they're not going to ask you this and leave it there. If you say whatever you say here, you have to explain. Why? It's obvious. The carbon dioxide given off by the worms during respiration will be taken in by the leaves for photosynthesis. Right? Remember, even the leaves themselves, they will also use up some of that, whatever carbon dioxide that they release, or rather that they use up here, is going to go to the worms. At the same time, the carbon dioxide that the worms give off will be taken by the plant for photosynthesis and for respiration. Because the plants must also respire. So there's a balance between the oxygen and carbon dioxide. Now, how does anaerobic respiration help us in industry? Yeast is used widely in biotechnology. That's a known fact. It is used in the fermentation of alcohols and the production of bread. Just to give you some examples, Here we are in the yeast can be used to make wine. Also, bread yeast is used to make the dough rise. Okay, that's when the carbon dioxide is released there. Next question. So that's a very small section that you have to know. You just need to say what it is used for. One is in the beer industry to make alcohol, and the other in the baking industry to help to raise the dough. You don't even have to say the second part is used in the baking industry. That's good enough. That's all you need to know in that part. Now, we're going back in our work. Define cellular respiration. A straightforward process whereby organic compounds are broken down using oxygen to release energy in the form of ATP. And remember, cellular respiration is a step-by-step process. That it doesn't happen in one bang. It's a step-by-step, and it's released in stages. Remember, what is respiration really speaking? It's burning food, using oxygen to burn food. But it's done in a very systematic way. Because if you take respiration, lots of books and lots of people will compare it to burning. Burning, what we mean by burning? If we take a match and we strike a match and we burn something. Oxygen is used in that process, and carbon dioxide is also given. But lots of heat energy is given. Whereas in respiration, there's a little bit of heat energy and a lot of ATP. So usable energy is used there. In the case of a fire, you're getting a lot of light and a lot of heat. No ATP. Okay, good. Move on. List the reactants. That's a lovely one. The reactants and the products of cellular respiration in both chemical equation and in words. And there we have it. We spoke about this already. 6O2 plus C6H12O6 will give us 6CO2 plus 6H2O plus energy. Or we can say in words, oxygen plus glucose gives us carbon dioxide, water, and energy. We can add here in the form of heat and ATP if you want to. But up to there, that's the definition. That's just an extension to what we're saying. Next question, also famous question. Tabulate the differences between respiration and photosynthesis. Respiration, first of all, I told you, where does it take place? Occurs in the cytoplasm and mitochondria. Photosynthesis takes place in the chloroplast. So that's your first difference, where they take place. Next difference, what is used? What is necessary? Oxygen is consumed. And what is produced? Carbon dioxide. That should be consumed. Okay? So not confused like I am, but consumed. So here oxygen is used or consumed, by the way, an easier word, used, and here carbon dioxide is used. Then, first of all, this one here is a catabolic reaction. Why? Because it uses energy. And I told you earlier, photosynthesis is an anabolic because it makes energy. What are the products? Remember what I told you. I told you at the beginning when you're tabulating the differences, where does it take place, what does it use, this is the extra one, and what is produced. Energy, carbon dioxide, water are produced. Or you can say, instead of energy, you can say ATP is produced. Whereas in photosynthesis, organic molecules are produced. Light is not essential for respiration. Any one of the phases, any phase, glycolysis, Krebs cycle, oxidative phosphorylation, light is not necessary. Whereas in photosynthesis, for the light-dependent phase, or the light phase, light is essential. So those are your tables of differences. Again, you could make their differences only if you understand the process. Next question says, compare aerobic and anaerobic respiration. Now remember, let's close that. The question did not ask you to tabulate here. I put these two questions deliberately next to each other so you know when I say tabulate, you put in a table. When I say compare, just like this, or explain the differences, then you just need to talk about it. Remember that. So firstly, let's look at the first one. Aerobic respiration occurs in the presence of oxygen and creates a maximum of 38 ATP. So two things we're talking about. When does it take place and what does it produce? While anaerobic respiration occurs in the absence of oxygen and creates a maximum of 2 ATP. Then we can go further. The products of aerobic respiration are carbon dioxide. We can go even further. Energy and water vapor. Right? While the products of anaerobic respiration are carbon dioxide, little energy, and alcohol or lactic acid. In plants, alcohol. In animals, lactic acid. So you can compare it in the same way that we compared photosynthesis and respiration, but now we're doing it without a table. Question 9. A little bit of reading again. The following diagram shows the apparatus used in an experiment. Nothing important there. Seeds of a pea plant were soaked in water and placed into 3 identical glass bottles. A 4th empty bottle was also used. Empty bottle. A carbon dioxide sensor was placed into each of the bottles. 1 bottle with seeds was placed into a freezer, the other one in a room, and the other in an oven. Answer the questions that follow. First question says, germinating seeds there, remember. What process is being investigated in this experiment? Again, because we're dealing with this section, it's easy. Cellular respiration. Write down an aim for this experiment. What do we want to do with this experiment? Aim for the experiment? Why are you doing this experiment? What do you want to show? To show or determine, or another nice word also, to illustrate. Illustrate the effect of temperature on respiration. Why? How does one get to this aim? How do we know that they're doing temperature? Yes, you got it. Because they told us the one was in a freezer, the one was in a normal room, and one was in an oven. So what was changing? Only thing that could be changing there is temperature. Unless somebody is lying to us. Okay? Is light necessary for this process? We just answered the question about that just now, and the answer would be no. Why was 1 bottle left empty without seeds? The 4th bottle. Notice this type of question in all investigative questions, it's coming. As a control, and also to know the normal amount of carbon dioxide in the air. Why was it necessary to soak the seeds in water? Why did we put dry seeds in? Now this, you needed to know a little bit other work. And that is so that the seeds start to germinate. Because if the seeds are not germinating, they are dormant. If they're dormant, no cellular respiration takes place. So this is why we have to do that. Now, the table below indicates the results of the experiment. Condition, no seeds, freezer, a room, temperature, oven. Amount of carbon dioxide in the air, given you all these possibilities. So they say, use the data to draw a graph. Remember when they're giving you groups of data like this, where there are different things doing different things, that is doing that, that is doing that, that is doing that, that is doing that. You can't draw a line graph to this. You can't even draw a pie graph because they haven't given you percentages of this. It doesn't even make 100%. To draw a pie chart, you must have something that totals up to 100%, and it must be giving you proportional. This is different things doing different things. So the best graph that you do here would be a bar graph. And there's our graph. Now notice when we draw our graph, all graphs must have a heading. That's the first one. The heading must have the independent variable and the dependent variable. Different conditions was what we changed. Whatever you could change, that's the independent variable. And because of this, there was different amounts of carbon dioxide in the bottles. There. Right, that's the first part. The second part, you must label your axes. Your x-axis is at the bottom here, horizontal axis, and your vertical axis is called the y-axis. Now, you're not getting a mark for putting x and y. We need a label there. And the label must come with units. In this case here, we're talking about condition. The condition doesn't have any units, so you don't use units. On the y-axis, we talk about the amount of carbon dioxide because we put our dependent variable on the y-axis and the independent variable on the x-axis. And notice, we gave the unit there in percentage. The next thing you have to do is to divide your lines. Now, you can use your lines in your book. And remember, look at the units that were given. 0,03, 0,04, 0,1, 0,03. So everything is decimal here. So you need to work in decimals. You start with 0,02, 4, 6, 8, 0,1, 0,1. So obviously, you needed to know a little bit of maths lit at least to answer a question like this, to understand that in decimals, this is how it's going to work out. So you have your values there put in. They must be equidistant. And notice, we're not using the figures as they are in this table and saying, right, okay, so we got 0,03, and then we're putting 0,04, then we're putting 0,1, then we put 0,03 again. We can't do that because if you look at a ruler, it doesn't go that way. It has fixed demarcations, and 1 centimeter is equal to 1 centimeter. Then on this side, on a bar graph, you've got to label each thing that you're putting there. That's number one. And notice that the bars, the width of all the bars, let's use pink, the width of all the bars are they're equal. And the distance from this bar to that bar and that bar to that bar are also equal. That is also worth a mark when you're doing the bar graph. Okay, and then you're plotting the graph. That's the easy part. You're plotting the graph and you're going to draw your columns that way. If you wanted to show something different, or in this one here, just one bar graph, it's easy. The things are already labeled. But if they give you two things to do, one set in an oven and one that you have to compare, then you'd have them together that way there. In terms of that, this is how you would draw your bar graph. So I'm hoping that that has helped you to understand. Now notice what we've done throughout in the questions. We've taken investigative questions. We've focused a lot on that because it's lacking. And in our explanation, we didn't go to the experiments, so we spoke about the experiments in the questions. At the same time, we looked at the skill of drawing a bar graph. We could then even give you this bar graph, and we could ask you from this bar graph, convert this to a table. We also looked at other questions of definitions, comparisons, tabulations, etc. Take that in. The skills that we've learned in this lesson is what should make you a better person. You are at point A, you must be moving further up. With that, I say goodbye, good luck, and God bless.